Are you here for the Myoglobin talk? Oh, okay. Yeah, so my name's Victor. I'm here to talk about myoglobin in this really interesting experiment. So, funny enough, myoglobin, which you usually hear hemoglobin, but myoglobin is actually basically the version of hemoglobin but for your muscles, so it delivers oxygen to your muscles. But what could be the bane of every weightlifter in existence? Guanidine hydrochloride. Yeah, I bet you've never heard of that. All right, moving on. Yeah, so what is guanidine hydrochloride? It's actually a denaturant used in a lot of biochemistry, and it causes the myoglobin to unfold. And if you notice, if you look at the myoglobin structure I have over here, there is something that we kind of see, and it's like a ring, and it's called the porphyrin ring. And we're going to be using that to actually measure how much myoglobin has been basically denatured by our guanidine hydrochloride. And the reason for that is this porphyrin ring right here, actually, because of the pi system, has a very distinct absorbance band, something called the sorbet band. Like the ice cream. Wait, no. Sorbet band. Sorry, it's not the sorbet band, it's the sorbet band. And this band is actually used to look at how much of the protein is present, because when there's more myoglobin present, you're going to see more absorbance in that sorbet band. And when there's less myoglobin present, that absorbance value is going to go down. Why? Because there's less myoglobin. In this case, because we're denaturing it with guanidine hydrochloride. So going into this lab, what we ended up doing is we had like 19 vials, and in it we put a mixture of a buffer, we put in the myoglobin, and we put in varying concentrations of guanidine hydrochloride. We kind of let that sit for around 20 minutes for the denaturing to happen, and then we just took those vials, put them in a cuvette, and stuck them into our UV vis, and then got our absorbance reading. And before I went to this lab, my hypothesis that I had was that as we add more guanidine hydrochloride, I kind of expected that at some point, like the absorbance value wouldn't really change. It'd be like a steady state. And the reason for that is once you've sort of knocked around, see all the myoglobin and it denatures, there's no more myoglobin to like break down. So in the beginning, you have one myoglobin and you have one guanidine hydrochloride, and smack each other and the myoglobin to associate. But eventually, you have myoglobin, and then you have a whole bunch of guanidine chloride that kind of just smack the myoglobin around, and then there's no more myoglobin, or there's so little that like the guanidine hydrochloride is like searching for myoglobin to break down, but there's nothing to break down anymore. So that's why I think at a certain point of guanidine hydrochloride concentration, the absorbance values will probably just peter off and kind of reach a steady state. And the second thing is, if you look at the equations over here for the theoretical and experimental fractional denaturing of the protein that we use to like predict that value and calculate those values, if you actually do some like magic with just not magic, it's just rearranging the equations using basically the fact that certain parts are equivalent to each other, you can find that the equations are actually really related. All right, moving on. All right, so now we're going to take a look at the actual results that we got from our Myo-Goblin experiment. Oh, oops, sorry. Myoglobin experiment, not goblin. So we, if you look here, we have our characteristic sorbet band here in our like low 300 to 400 nanometer wavelength region, and the little diamonds are the max absorbance values for each sample that we checked. And we have those values over here if you'd like to check. But if you notice, we have a cluster of max absorbance values at the very top and a cluster at the very bottom. And those are indicative of the fact, and it's indicative of the fact that as we add a little bit of guanidine hydrochloride, not much of the myoglobin really denatures until it fully starts to denature, which is what you see this progression in the middle is. And then at the very bottom, all the values are a little clustered together and they're all more or less the same. So it's like we have a steady state in the beginning when we slowly add guanidine hydrochloride, and then we have a steady state at the end where adding more guanidine hydrochloride doesn't really cause any more denaturing of protein. And we can also use this data to find out what's the likelihood that myoglobin we denature when there is no denaturant. And to do that, we're going to go to our next graph. In the next graph here, we graphed the max absorbance values of each respective concentration of guanidine hydrochloride. As you can see, illustrated very clearly, we have our steady state on the left and our steady state on the right. And in the middle here is where we have our change. So the next thing we're going to introduce is we're going to take the max absorbance when there is no guanidine hydrochloride present. So now we're going to have our AN value, which is 1.03 when there is no denaturant present. And then we took the minimum value for when all the protein has been unfolded, and adding more guanidine hydrochloride doesn't really do anything. And we took that value to be 0.551. It was the most minimum value, so we could have as many values as possible. And as a result of that, we were able to calculate the KEQ, the equilibrium constant, using this equation, A representing the absorbance at that specific concentration value. And then taking that equilibrium constant, we're able to find the Gibbs free energy of the denaturing process using the equation here. And, you might be wondering, what do we do with these values? What are these values are relevant? And we go back to the previous graph right here. We can't really get anything from the steady state up here to the steady state at the bottom, but we can get values from the steady state. And when we individually graph the steady state, so these orange values here, so going from this, we can see that it is a linear relationship. And from it, we can get M, which is the slope of how much the Gibbs free energy of the unfolding process or the denaturing process changes as we increase concentration. CM is the concentration of guanine hydrochloride that we get an equal amount of normal myoglobin and unfolded myoglobin. And then finally, the Gibbs free energy of when it is just in water and the likelihood that it would denature on its own. And as you can see, that ends up being the positive 25,000 joules per mole. It's very unlikely, as you can see, that myoglobin would just spontaneously denature on its own. And using the slope, you might wonder, 25,000 joules per mole, can you get anything about how fast the reaction proceeds? And the answer to that is no. You can't really get anything with Gibbs free energy about how fast the denaturing will proceed. You can't really pull kinetic information for like time from spontaneity values like this. So these values tell you that, when delta G is 25,000 joules, it's very unspontaneous. It doesn't really give off energy to denature, so it won't happen. And this value of change doesn't really tell you if the reaction will happen quickly or not. And the next thing, CM, what's that concentration? We find it to be around 0.997 molarity of guanine hydrochloride. And going off of that, we can then calculate how much protein is dissociated, both experimentally using our KEQ values and theoretically using this equation over here. And when you graph those two values, we can find, approximate what is the concentration when both the unfolded and native normal myoglobin are at. What's that concentration to get half and half denaturing? And we actually find it's around 1. Which, if you remember, our CM is practically 1, 0.997. That's more or less 1. And look, our experimental value says it's about 1. That's very interesting. But if you look at this, we graph our theoretical fractional association over here in our blue line, and then our orange dots are experimental values. And you can see they match up pretty close. And actually, when you find the chi-square, it's a pretty small chi-square value. But when you decide to use some magical solver functions from Excel, you can get this value to be a little bit better. It's 0.0421 instead of 0.0424. And you see the values match up slightly better, but it isn't like a significant difference that makes it all that much better. So you might be wondering what this all means, right? What are the conclusions we can take from this? Number one, if you want to build muscle and hit a PR, don't drink guanidine hydrochloride. Anyhow, but based upon that, like answering my hypothesis is like my hypothesis ended up being more or less supported by the data, and it's just that the F theoretical, fractional denaturing theoretical, and experimental did match pretty well. It wasn't perfect, but I would trust the theoretical model to tell me what would happen if I added guanidine hydrochloride, because it just matched super well. And the other thing was, yeah, we did see that steady state at the end. And, that just shows you that adding more doesn't necessarily mean it's going to work better. Usually, as you notice, as we added more, we did get more denaturing. But at some point, adding more in excess doesn't really help you. Things that, you can think about, takeaways from the lab, I suppose, besides don't drink guanidine hydrochloride, is, there's a lot of things that affect whether or not the cells and functions of those cells in our body work. And better understanding how certain molecules interact, or environmental things like pH and temperature, etc., interact to affect those cells is really important, just for both our own health and health medical usage in like hospitals, etc., and also for scientists and researchers, so we know more about human physiology. And maybe you'll be inspired by this lab experiment and go do more studies on human phys and human myoglobin. But that's up to you. It's been a great time. I've had a lot of fun, and hope you have a great day. Thanks for listening. Goodbye.